Hello, everybody, and welcome to this discussion on telomerase. So telomerase is a large ribonuclear protein. It functions in the addition of telomeric repeats to linear chromosomes, facilitating the replication of the 3' ends of such chromosomes. You have to also keep in mind that telomeres are structures which maintain the genomic integrity of linear chromosomes. For instance, telomeres, along with associated proteins, facilitate the distinguishment of the ends of chromosomes from double-stranded breaks by DNA repair machinery. Furthermore, shortening of telomeres to a sufficient degree results in two potential consequences. It can lead to telomeric fusions, and it can also lead to cell senescence. What you have to keep in mind with regards to telomerase is that it's composed of two primary components. It's composed of the telomerase reverse transcriptase, or TERT. This is the protein component. It's also composed of telomerase RNA, or the TR component, which is the RNA component. The protein component, or TERT, is relatively conserved across species. However, TR, or telomerase RNA, is highly divergent across species, so you have to keep this in mind. Telomerase RNA is highly diverged across species, whereas the protein component of telomerase, that is the TERT component, is relatively conserved across species. So let me talk about TERT, or the protein component of telomerase. It may contain the following functional domains. It may contain the TERT N-terminal domain, or TEN. The reason I say may is because some TERTs from certain organisms may contain some but not all of these domains, but it contains the TERT N-terminal domain. This is the TEN domain. It also contains the TERT RNA binding domain, or the TRBD. It also contains reverse transcriptase domain, or the RT domain, and it also contains a C-terminal extension. Now, the TEN domain interacts with single-stranded telomeric repeats. On the other hand, the TRBD domain interacts or binds sites on the telomerase RNA. And then the reverse transcriptase domain, or the RT domain, and the C-terminal extension both function in catalyzing the addition of telomeric repeats to the 3' end. It's good to know that telomerase RNA has certain pathological links if there are mutations within the telomerase RNA sequence. So mutations in telomerase RNA are linked to various pathologies. These pathologies include dyskeratosis congenita. They also include myelodysplasia and aplastic anemia, as well as idiopathic pulmonary fibrosis. And what you have to keep in mind is that when we say that telomerase RNA is highly divergent across species, we mean that it is divergent with respect to sequence as well as length. So such that sequence lengths are the following. For ciliates, the sequence lengths of telomerase RNA is roughly 147 to 205 nucleotides. For vertebrates, it's roughly 312 to 515 nucleotides. And in yeast, it can be anywhere from 779 to over 2030 nucleotides in length. And what you have to keep in mind here is that the secondary structure is relatively conserved. Or to be more accurate, I should say that secondary structure conservation can be observed. And this makes sense given that telomerase RNA interacts with the relatively conserved structure of TERT. So TERT is relatively conserved across species, so it would make sense that the telomerase RNA secondary structure has some conservation to it, such that it can interact with what is essentially a conserved or a relatively conserved TERT protein structure. Furthermore, this information tells us that conservation of secondary structure RNA structure is indicative of functional roles for respective motifs in that secondary RNA structure. So what I mean here is that the fact that there is conservation in the secondary structure of RNA should tell us that there is function that relies on that conservation of the secondary RNA structure. So we said that the secondary structure of telomerase RNA has some conservation, whereas the sequence length and the sequence itself is highly divergent. Here, it's good to know where the conserved RNA structures or what the conserved RNA structures include. So you should keep those things in mind. These are conserved structures in telomerase RNA. They include the loop, which contains the template sequence of telomerase RNA. They also include the 5' template boundary element, as well as the stem terminus element, or the STE, and the loop closing helix. Here, it's noteworthy to examine the secondary structure of vertebrates' telomerase RNA. So let's look at the telomerase RNA of vertebrates. The telomerase RNA of vertebrates contains 4 conserved domains. These 4 conserved domains are the pseudoknot, which contains the template for telomerase synthesis of telomeric repeats. It also includes a conserved regions 4 and 5. So conserved regions 4 and 5 are also labeled CR4, CR5. Now, both the pseudoknot and the CR4, CR5 domains bind to TERT independently and comprise the catalytic core of telomerase RNA. Also, some of the conserved domains that are left include the BOX-HACA domain and the CR7 domain.